kind of lecture, you should know what is the genetic and basis, discuss basis, inherited and acquired disorder. You can list the clinical presentation of few genetic disorders. You will be able to explain recent advancement in the field of diagnostic technology, techniques and molecular areas with some example in its basis. Our healthcare system is critically and crucially dependent upon diagnostic. As today's medical decision making is strongly based upon the diagnostic result. Right from the genetic test that can help notify personalized cancer treatment to microbial culture for recognition of right antibiotic combating an infection, diagnostic provide critical insight at every stage of medical care prevention, detection, diagnosis, treatment, and successful management of health condition. The main categories of diagnostic are clinical chemistry, immunology, hematology, microbiology, and today that we are discussing the molecular diagnostic. The molecular diagnostic has attracted particular attention in recent years owing to deep insight it brings diagnosis and treatment. Molecular diagnostic has transformed diagnostic dynamically, leading to insight in research and treatment in many disease states that are revolutionizing healthcare. Today class will provide an overview of the current scenario of molecular diagnosis with glimpses of the key technology that are driving the molecular revolution with pinpointing some specific disease and disorder. In this slide that is showing the old way and the new way. When we were the student in that time, we study the pathology was at the microscopic level. The tissue is taken, biopsies, and then go into a process, a slide is made, and that diagnosis is made on the microscope. Now, in the recent way, the patient tissue sample is taken, then proteomic, genomic, the DNA is obtained and observed by different techniques. So, now the things go at the molecular level for the diagnostic purpose. See, this is a cell, the diagrammatic view, inside which there is a nucleus. So inside the nucleus there is a chromosome. That chromosome is a DNA. Inside that chromosome that we call it as a gene. And that gene having a certain protein basis. The whole genetic information about any person lies inside the DNA. The typical human cell contains about 46 chromosomes, having a 23 pair each, and one each homologous pair of autosome that we call as a one pair of sex chromosome, either XX or XY. As we all know, the gene is made up of RNA and DNA, nucleic acid. RNA is present in the nucleus, but DNA is present both in nucleus as well as mitochondria. These nucleic acids are made up of nucleotide, whose composition include nitrogen bases, a sugar, sugar deoxyribose in DNA and ribose in RNA, and a phosphate group. This is the whole that you have got a previous knowledge. You have to recall your knowledge. So the nitrogen base can either be purine, which contains iodine or guanine, or pyrimidine, that is thymine, cytosine, or uracil. The purine binds always with pyrimidine, that is complementary. As we know, the human germ cells, ova and sperms, both contain 23 chromosomes, that is a haploid number or 1n. While all the nucleated somatic cells of the human body contain 23 pair of chromosomes, that is we call it a diploid number of 2n. So that 2n, that means 44, that chromosome will be autosome, or two sex chromosome being either a female, then XX, in case of male, it will be a 46XY. Cytogenic is actually a branch of science which deals with the study of this human chromosome abnormality. called cytogenetics. When we are talking about the genetic disorder, then we classified into as a mutation either in a single gene, either the chromosomal disorders, either the multigenic disorders. So mutation in a single gene is usually highly penetrant, that is mutation typically result in a disease and follow classic Mendelian inheritance pattern. Chromosomal cytogenic disorders, these arise from structural example break or numerical alteration in chromosome, either to say trisomy 21 or Edward 18. So any chromosomal abnormality, they are usually highly penetrant. And multigenic disorder or multifactorial disorder, most common cause of genetic disorder, and are caused by the complex interaction of the multiple variant, not mutant form of gene, polymorphism, and environmental factors that are involved. See, this is the structure of the chromosome that you are seeing in the picture one chromosome, and second, you are seeing one portion is transferred to other, and second from that another form, that is called translocation. One arm is deleted, that we call the deletion, and sometime below portion goes up portion comes, that we call inversion. Sometimes this chromosome take a ring like this, that is a ring formation, and sometimes both the things get same, that is called an isochromosome. So whenever there is a mutation, mutation means complete change. So either it is a single nucleotide base substitution, or it may be an insertion or deletion of base leading to alteration in reading frame of DNA strand. That type of mutation we call as a frame shift mutation. Whenever there is a single nucleotide base substitution, that is called a point mutation. Some amino acid, for example, of as a point mutation, some amino acid is formed even by the change codon, that is called a silence mutation, or the replacement of amino acid by another, like in missense in sickle cell, where glutamic acid is replaced by valine. Replacement of amino acid by a stop codon, that we call a UAA and UGA and UAG. That is called a nonsense mutation, and that type of mutation usually occur in a beta thalassemia. So genetic, when we are talking about the genetic disorder, as already I told you, the classification either on single gene or the multifactorial or the chromosomal. So single gene disorder always observe the Mendelian inheritance pattern, so in which the things come either they are autosomal dominant, autosomal recessive, or the X-linked dominant or the X-linked recessive. When the environmental factor that come under the heading of multifactorial, in which there is the involvement of the gene is there along with the environment, that is diabetes mellitus, hypertension, gout, coronary heart disease. When we simply talk about any deletion or addition in that chromosome occur, either that monosomy or trisomy, that is. So transmission pattern of single gene disorders, either they are autosomal dominant, either they are autosomal recessive, either they are X-linked disorders. These are the few examples of autosomal dominant disorders in which von Willebrand disease, familial adenomatous hypercholesteremia, adult polycystic kidney, dystrophia myotonica, osteogenesis imperfecta, Marfan syndrome, intermittent porphyria, neurofibromatosis, achondroplasia, neurofibromatosis, tuberous sclerosis, Huntington disease, hereditary. So these are all the examples of autosomal dominant. Some important autosomal disorders, if we discuss of the Marfan syndrome, actually it is an autosomal dominant disease having a mutation in the fibrillary gene. So on the chromosome 15q21. Fibrillin behave as a scaffolding protein for alignment of elastic fiber. So any defect affecting the following system, they affect on the skeletal, gene effect on the skeletal defect. So person get a tall and thin build, long and slender finger, hyperextensive joints, inward depression, sternum, that is a pigeon deformity chest. On the cardiovascular, it will get a mitral valve prolapse, medial degeneration causing dissecting aortic aneurysm or aortic regurgitation. In the ocular changes, either there are bilateral dislocation or subluxation of the lens, that is we call as a ectopia lentis. That all things are happening in the Marfan syndrome. This is which is the autosomal disease. If we talk about the neurofibromatosis, they are again on the basis of gene, either NF1 or NF2. NF1 also called a von Wreckling disease, and NF2 also called as a bilateral acoustic neurofibromatosis. So NF1 is more common, that is about around 90%. Presence of neural tumor of neurofibroma in the body, which can be cutaneous, subcutaneous, or placiform. 6 or more pigmented skin lesion called capsuloid spots present on the skin. Pigmented iris hematoma called Lish nodule also present. Associated skeletal muscle defect like scoliosis, bone cyst, or tuberous orthosis usually occur. And risk of developing meningioma, pheochromocytoma, and Wilms tumor. Here, in case of neurofibromatosis 2, that is less common and seen usually less than 10% population. Bilateral acoustic neuroma usually occur. Multiple meningioma tumors occur in the brain. Capsulate spots are present, but Lish nodules are here in this case are absent. If we are talking about the Ehlers-Danlos syndrome, it is an inherited tissue disease due to defect in a collagen structure or synthesis. So rest of the features are described over here. So this Ehlers-Danlos syndrome, type 3, type 4, and type 5, all are written here. So autosomal recessive inheritance disease. When we talk about the autosomal recessive inheritance disease, usually it includes most important area of metabolism. Either it have a complete penetrance, which is more common, and onset usually frequently in early life. Mutation are rarely detected clinically until several generations have passed. This is an example of autosomal recessive disease that you're seeing. Friedreich ataxia, phenylketonuria, galactosomia, cystic fibrosis, hemochromatosis, antitrypsin, alpha-1 antitrypsin deficiency, Wilson disease, alkaptonuria, sickle cell anemia disease, thalassemia, homocystinuria, muscular atrophy, adrenal hyperplasia. Now coming to talk about the X-linked disorder, all the sex-linked disorder are X-linked and most are recessive. They are fully expressed in male because mutant. So X-linked disorders means female are the carrier, male are affected one. When we talk about the chromosomal disorders, then a cytogenic disorder may be due to alteration in the number of and structure of the chromosome. Karyotype are given as the total number of chromosomes followed by the X chromosome complement, and then abnormality in ascending numerical order. Example, male with trisomy 21 is designated as 47XYP21. So see, this is a picture of the that you are seeing karyotyping, in which these all the 46 chromosomes you are seeing, 22 pairs, and 23 is either X chromosome or the sex chromosome or the Y chromosome. So in which you are seeing regions, arms of the chromosome that showing structure. See, this is one thing. As you go from 1 to 2, 3, the size of the chromosome gets smaller and smaller. These chromosomal disorders, when we're talking about, they come under numerical disorder. Monosomy associated with one less normal chromosome. Trisomy means associated with one extra chromosome. Mosaicism associated with one or more population of cells, some with normal chromosome complement, or other with extra missing. Cytogenic disorders usually involving autosomes, that is trisomy 21, that we call as a Down syndrome, trisomy 18, Edward syndrome, trisomy 13, that we call as a Petu syndrome, chromosome 22q11 to deletion syndrome. See what happened in the trisomy Down syndrome that we call as the trisomy 21, it incidence 1 in 700 birth. So trisomy 21 type is having a 47XX plus 21 chromosome. So plus 21 something added at the 21 chromosome. So here the translocation type that you are seeing in the picture. So the person who suffer from this Edward syndrome, having is a highly mentally retarded, having a abundant skin, congenital heart defect, and intestinal stenosis, umbilical hernia, hypotonia. Such type of patient are more predisposed to the leukemia. If we talk about the Edward syndrome, that we call as a trisomy 18, and this usually occurs 1 in 8,000 birth, and trisomy 18 type is 47XX plus 18 chromosome. So here microophthalmia, polydactyl with fingers, cleft lip and palate, microcephaly and mental retardation, rocker bottom feet, usually find in this Edward syndrome. If we talk about the trisomy 31, that is Petu syndrome, this usually occur 1 in 15,000 birth. So trisomy 13 type is 47XX plus 13 chromosome. In this microphthalmia, polydactyl, cleft lip palate, mental retardation, renal defect, umbilical hernia, cardiac defect, and rocker bottom feet. Cytogenic disorder involving a sex chromosome that we are, I told you, 22 are the autosomal chromosome and 23rd is a sex chromosome, either X or Y. They are involving Klinefelter syndrome in two or more X chromosome and at least one Y chromosome, like 47XXY. Turner syndrome, complete or partial monosomy of X chromosome, either 45X or with partial deletion of the X chromosome, and mosaicism, that is 45X and 46XX. Hermaphroditism and the pseudo-hermaphroditism that also comes under the heading of genetic disorder of the sex chromosome. See, in the Klinefelter syndrome on the right side that you are seeing, there is a karyotype in which this gold rounded circle is showing that this X chromosome high is affected and portion is detached. So this usually occurs in male and detected at the puberty. The main features of this are the small testes, person having a lactosterol level, gynecomastia, micropenis, reduced facial and body hair, and infertility. It leading disability and delayed speech and language development is usually 1 in 500 to 1,000 normal male birth. Extra copy of X chromosome in each cell. Case by non-restriction of XX homologous. When we are talking about these genetic disorders, so there should be a proper evaluation. The first evaluation that can be done, prenatal evaluation on the basis of their previous history. So that is performed on the fetal cells which are obtained either by means of amniocentesis or by chronic villi biopsy or from the cord blood. Since amniocentesis is done at the gestation of the 14th week and the chronic villi biopsy as early as we can do at the 8th to 10th week, and even from the cord blood we can make early diagnosis of the prenatal evaluation. So prenatal evaluation usually is suspected if any female having pregnancy at the 35 or more, parent with structural chromosomal abnormality, previous child with chromosomal abnormality, carrier of an X-linked disease to determine the fetal gender. Prenatal evaluation and the postnatal evaluation is usually performed by peripheral lymphocyte. If we are suspecting multiple congenital anomaly, unexplained mental retardation, suspect chromosome abnormality like Down or Prader-Willi syndrome, suspected Fragile X syndrome, infertility to rule out sex chromosome abnormality, recurrent abortion. So these are the few indications in which we also do the postnatal evaluation to make a proper diagnosis at the molecular level. So indication for acquired genetic alteration, sometimes this mutation occur at any stage and leads to the malignancy. Specific mutation or cytogenic alteration that are hallmark of certain tumors, example BCR and ABL in chronic myelogenous leukemia. Determination of colonity as an indicator of malignancy. Indication of genetic alteration that can be influence therapy, example EGF receptor mutation in lung cancer. Determination of treatment efficacy, presence or residual disease. Detection of therapy-resistant mutant of given tumor. Diagnosis and management of infection disease also can be done at the molecular level, like HIV or mycobacteria. Identification of specific drug-resistant microbes. Determination of treatment efficacy of the residual virum like loads in hepatitis C. Now coming on the techniques that are applied at up till what we have discussed, inherited genetic disorder, acquired disorders, and due to environmental factors, some disorder occur. How we can lead to the diagnosis. So this diagnosis depend upon the common method used in on these molecular diagnostic techniques. Uh, uh, Used in the molecular biology, biochemistry, chemistry, and biophysics, which generally involve manipulation and analysis of DNA, RNA, and protein. These techniques have utility in virtually every area of diagnostic pathology, neoplastic disorder, infectious disease, inherited condition, and identity determination. So, as a broadly, we classify these molecular technique as the amplification technique, PCR, and so many its modification comes. So that technique comes under the heading of amplification technique. Another techniques that we call as a blotting technique, in which the southern blot, dot blot, uh, RFLP, refractive fragmental, and says northern blot, western blot. In hybridization technique, in situ hybridization, fish and its modification like microarray and other techniques. So one by one we will discuss. Coming to the amplification technique, so polymerase chain reaction, which is developed by the Carey Mullis in 1984, test tube method amplifies a selected DNA fragment. So it amplifies a selected DNA fragment which we want to make into amplify. So for that we need a DNA primer, Taq polymerase enzyme. So two set of primer, generally 20 to 30 nucleotide long, synthetically produced, 46 of CG content preferred. PCR cycle. In first step, there is a denaturation of DNA that usually done at the 94 degrees centigrade, in which the DNA is denatured and separated. Second, that is an annealing with phase, which done with the help of primer. The temperature is lowered down up to 55 degrees, in which uh, the DNA gets separated and attached with the and primer is. Then the next step in the PCR overall is the extension of the primer at the by increasing the temperature up to 72 degrees C with the help of enzyme with substrate. So it results in two copies of target sequence. So this is the modification that now comes: real-time PCR, RT-PCR, nested PCR, multiplex PCR, inverse PCR, asymmetric PCR, encode PCR, immuno-PCR, competitive PCR, in situ PCR, single-cell PCR, ligase-chain PCR, amplification of refractive mutation system, Q-beta replicase, nucleic acid shrinkage based on amplification. So these are all the modifications of the PCR recent days came. Reverse transcriptase PCR that amplify DNA from RNA. Here the enzyme which are used, that is called reverse transcriptase thermostable enzyme, in which from single copy we get a multiple copies of the PCR DNA. Actually, real-time PCR is a quantitative PCR in which we this quantity or the viral load or other things we see. So in the real-time PCR, while the reaction is occurring after each cycle of the PCR. Nested pre-PCR, in this use a two set of primer, outer and inner, increase both sensitivity and specificity of the assay. So this is more sensitive and reliable test than the previous PCR. Multiplex PSR use more than one pair of primer, application of many targets in one reaction. So this is a multiplex PSR in which amplification of many target in one reaction. In previous one, there is a one target which get amplified. Here, multiple targets that we call a multiplex PCR. What are the advantages? They are automated, they are rapid, they are reliable, content, they are sensitive and well defined. Now, after PCR, another technique that we use for the molecular level is the blotting technique. So this blotting technique is used for the identification of specific DNA, RNA, or protein from thousands of each. So in this type blotting technique, southern blot that we use for the DNA purpose, northern blot for the RNA purpose, and western blot for the protein purpose. Southern blotting technique is based on the specific base pairing rules of complementary nucleic acid strands. Here, in the dot blot technique, sample is directly applied to a slot on a specific blotting apparatus containing nylon membrane. Restriction fragment length polymorphism. This is used in inherited difference in the pattern of restriction map produced by the digestion of a specific restriction endonucleus. So DNA variation that result in restriction fragment length polymorphism, either it is a single nucleotide polymorphism, variable number tandem repeats, or in case of mutation. Uses of this refractive fragment length polymorphism in tracing chromosome from parent to offspring, prenatal diagnosis of disease like sickle cell anemia, phenylketonuria, for the purpose of medical legal, detect point mutation, insertion, and a deletion by means of northern blot technique. Principle here, RNA and DNA hybridization technique. See here the technique is used, the sample is taken, RNA is excreted, and on the RNA is separated by size by means of electrophoresis. Then by this northern blot technique, transfer RNA to a membrane. Then RNA fixed to membrane with ultraviolet light is given. So membrane hybridized with label probes. So hybridized with label probes. So after that, there are visualization of label probe on an X-ray film. Western blot technique, antigen antibody interaction. So antigen protein of interest and antibody on the probe. So again in Western blotting technique, what happened? Antigen sample is put They are separated by the protein and the separation gel, and then they are putting on the blotting tank. Protein transferred to nitrocellular sheets. Then they are they are separated immunostaining slots. Then they are put on the autoradiography, develop and fix autograph. Then these antigen beds are visualized by this technique. Another technique that is called fish test or a fluorescent in situ hybridization. So in this, at the stage of the metaphase, chromosome or fluorescent probe use a very sensitive test. See, probe of DNA level with a fluorescent molecule, it denature, and then they hybridize and they observe under fluorescent microscope. What are the application of fish? Whenever we are suspecting aneuploidy for its diagnosis, demonstration of any small portion or subtle microdeletion on the chromosome, detection of complex translocation, analysis of gene amplification, either HER2 new positive, HER2 new mic, mapping newly isolated genes. Multicolor fish is a spectral karyotyping. Combination of 5 fluorochrome are used. By accurate computer generated signal, the entire human genes can be visualized. This is a picture of multicolor fish. Another advanced recent technique that we call the microarray technique. So here, this is used for the genotyping, for the gene expression. So here in this microarray DNA chip uh, is at the next generation sequencing is done, contain thousands of known immobilized DNA sequence. The fluorescent tag samples to be sequenced is added. There are a lot of applications of molecular diagnostic technique to make a proper diagnosis. Say in hematological malignancy, this is the molecular level. In chronic myeloid leukemia in epilepsy, there is a translocation usually occur in 922 level, and then follicular non-equivalent form is occur. So whole of our management depend upon the diagnosis, and now the diagnosis depend on these these molecular techniques where we can find out this chromosomal and mutations and so on. See, this is a normal and this is the abnormal translocation that you are seeing. So these molecular tests are also used in the certain infectious disease to make diagnosis in case of viruses like herpes simplex virus, HCV, HIV, EBV, cytomegalovirus, HBV, in case of bacteria, in case of parasites, in case of fungi, in case of inherited genetic disease like albinism, alpha-1 antitrypsin deficiency, telangiectasia, cystic fibrosis, familial hypercholesterolemia, hemophilia A and B, osteogenesis imperfecta, maculocytosis, sickle cell anemia, thalassemia, von Willebrand disease. So thank you for paying attention